The following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
I'm going to talk about what we call the genetic code. And I want to pick your brains for a second over here. Now, when we as biologists began to suspect that the instructions for proteins were contained or encoded in DNA, we immediately began to figure out that there was a problem. Now, we only have 4 nucleotide bases, and you should know them. We have adenine, thymine, cytosine, and guanine. But on the flip side, we have 20 different amino acids. And I just told you that the sequences of these bases, these nitrogenous bases and nucleotides, are used to determine the formation of these amino acids. So, for instance, if one nucleotide coded for one amino acid, we would have a shortfall of 16 nucleotide bases because we would only have 4 nucleotide bases to do this job. So it's right off the bat not logical to assume that one nucleotide dictates the formation or the addition of a specific amino acid because those numbers don't add up. Now, so the question I'm trying to bring out, or what I want you guys to think about, or what I'm trying to encourage at this point in time, is for you to ask yourself this question. How many nucleotides do you need to specify one amino acid in a protein chain? How many nucleotides do you need to dictate one amino acid in a protein, in a polypeptide chain? What if we took these nucleotides we have, for instance, adenine and thymine, and we put them together as a pair? Or, for instance, we combined guanine and thymine, and we use those combinations as a code to direct the synthesis of amino acids. That would mean we'd have, since we have 4 nitrogenous bases, we'd have 4 to the power 2 possible combinations of these, which would mean we have 16 possible combinations. If we're combining those nucleotides, that's still less than 20. That's still less than 20. Remember, we're trying to make 20 amino acids. Okay, so that doesn't work either. So this brings me to what we call the triplet genetic code. I'm going to highlight that, the triplet genetic code. It just so turns out that nucleotide triplets, or triplets of nucleotide bases, are the smallest individual units that can code for a single amino acid. So what do I mean by this? So if you have an amino acid being formed or being added to a polypeptide chain in translation, the instructions dictating that that amino acid is added came from a combination or a tandem of 3 nucleotide pairs, what we call a nucleotide triplet, or a triplet of nucleotide bases. So if 3 consecutive nucleotide bases code for one amino acid, that would mean that we have actually 4 to the power 3 possible combinations of those nucleotide triplets, which is 64 possible combinations. That is way more, way more than 20. So that should suffice as far as making sure that we can code for every single individual amino acid. And this is what we refer to as the triplet code of genetics, or the triplet genetic code. In the triplet code, the genetic instructions that direct the synthesis of a polypeptide chain are actually written in DNA as a series of non-overlapping 3 nucleotide units that we call codons. So what a codon is, it's a tandem or a series of 3 nucleotides back to back in DNA. So 3 nucleotides will make 1 codon, and 1 codon will code for 1 amino acid. So I hope this is starting to make sense, and that's why we call it the triplet genetic code, because we have 3 nucleotides making 1 codon. So 1 codon will direct the addition of a specific amino acids in a polypeptide chain. Now, in mRNA, a codon will represent a series of 3 nucleotides, like I've said. So one thing I want you to make sure you take away from this slide is that 1 codon will code for 1 amino acid. It's 1 to 1. But that codon consists of 3 nucleotide bases. So it's a triplet unit, and this is why we call it the triplet genetic code. And I've repeated that a number of times but I hope that with this constant repetitions, this concept is starting to stick in. Now, during the process of transcription, a gene will determine the series of nucleotides in the RNA transcript. So I've already explained that. A gene will direct the synthesis of RNA, messenger RNA, in the process of transcription. So for each gene, only one of the two DNA strands is ever transcribed. Okay, so, and only one of the two DNA strands is ever transcribed, and the same strand is used as a template for the same gene always. Now, during translation, which is the second part, as far as expressing these genes is concerned, the sequence of codons on mRNA is then translated into a sequence of amino acid. So what this slide is meant to make you understand is that the genetic code is a little more complex than you would expect. And what I mean by that is that for you to code for one amino acid in a polypeptide chain, you need a combination of 3 nucleotides, which is called a codon. So to give you an example, if I had an mRNA strand that had about 300 nucleotides, the protein that it would code for would have 100 amino acids in its length. I hope that makes sense because of the simple fact that 3 nucleotides, a triplet, codes for 1 amino acid. And for that reason, an mRNA strand that was 300 nucleotides in length would code for a polypeptide that was only 100 amino acids in length. So that's what we mean by the genetic code. And the diagram on the right there gives you a really good representation of what I'm talking about. So for instance, AUG, adenine, uracil, and guanine in RNA, that's one codon. ACG, adenine, cytosine, and guanine, that's a different codon. So codons are basically nucleotide triplets of nucleotides, and each of these codons will code for a specific amino acid in the process of translation. So let's move on. Now,